Hi there, Chem 6B students. I hope you all are doing well. In lieu of actually doing the experiment in person, because we still can't do that yet, I wanted to put together a demonstration to illustrate what we would have been doing in lab. And it turns out this week's lab on enzyme catalysis is actually fairly simple and even safe and legal enough to do at home. So I put together the materials and methods to do it at home, and I'll be posting those as a separate document on Canvas. But first I wanted to just show you what I did at home in order to do this lab exercise. So first, this lab is designed to get you all thinking about enzymes a bit more and to be getting some hands-on experience with the topics and materials that were covered in lecture. Mainly this lab exercise is to get you thinking about the substrate specificity, so the fact that enzymes recognize specific molecules or substrates. They act on very specific molecules. They don't just act on any and every molecule. And then also to be thinking about the importance of protein or enzyme three-dimensional structure on activity. So proteins, enzymes have to have specific structures, particularly at the active site where the chemical reaction occurs, in order for an enzyme to be catalytically active. Now, the materials you'll need in order to carry out this experiment are relatively few and simple. You can do it with most things that would be lying around your kitchen. A few things you'll need that you probably don't have in your kitchen, though, are going to be lactase tablets, some glucose test strips. You can probably use any sort of test strips that are used for urinalysis. You can find those at most drugstores, although I was having some difficulty. So I ended up just buying what are called diasticks, and they're produced by the company Bayer. They're designed specifically for testing glucose. And then you'll also need some table sugar and milk. That could be any kind of dairy milk, from skim milk to whole milk, 2%, 1%, whatever your choice is. So make sure you have those things on hand, as well as the other things that are listed in the materials. So the first thing you want to do is take your lactase tablet, and you want to crush it up so that it will dissolve in water better. So I ended up just taking a butter knife, cut it into several pieces, and then I used that flat edge to just smash it into a powder. And then I took that lactase tablet powder and put it in some water to dissolve it. And you'll note, probably regardless of the brand you buy, it's not going to dissolve completely because the tablet isn't 100% lactase enzyme. It has several other ingredients. Oftentimes these include cellulose. Now cellulose, you've learned about previously. It's a polysaccharide that comes from plants. So cellulose is not soluble in water. So don't worry, the lactase is probably dissolved in the water. If you see some stuff floating around, if you decide to do this at home, it's probably just mostly cellulose that's just not dissolving in water. So just give it a few minutes. Put it in the fridge when you're not using it, just to keep the enzyme stable. But the enzyme portion should be all dissolved in the water at that point. Now, what I just prepared, the lactase solution in water, will serve as the active enzyme control. And the other thing you want to make is your heat denatured enzyme sample. And so you can do that a variety of ways, or using a variety of containers anyway. What I did is I just took about half of the lactase solution that I had just made, and I put it in a glass jar, and then I put it in some boiling water on my stove for 15 minutes. That's long enough. And then once that is done, you'll want to take it out of the boiling water, and you can just let it cool down to room temperature on your kitchen countertop. You can transfer it to another cup first. But in the end, I ended up just transferring it to a new cup, which, as you can see, I have labeled as the denatured lactase in this yellow cup here. So the next thing you want to make sure you have is going to be your substrate samples. So the first is going to be lactose, which is found in milk. Since I don't have just lactose powder hanging around, but lactose is also known as milk sugar. It's prevalent in milk, so you can use milk of any kind. The other substrate that we'll be using to be investigating substrate specificity a bit is sucrose, which is also table sugar or cane sugar. I would suggest using white sugar. It dissolves better in water as compared to brown sugar. So you can just take some table sugar. What I did is I took, well, actually, the methods say to dissolve a gram in 20 mils. 1 teaspoon is actually about 4 grams. So what I did is I took 1 teaspoon and dissolved it in 80 mils of water, which is about 1/3 of a cup of water. And then if you're like me and you had an enthusiastic lab assistant, that person can help you dissolve it just simply by stirring in water. It will go into solution very quickly, and then you'll have your sucrose solution ready to go. Now at this point you're ready to begin the experiment. Instead of using test tubes, since I don't have those lying around at home, I actually just used an ice tray for my different samples. So as the methods indicate, there are 6 samples, A through F. Now A through C are going to have the active lactase solution, wasn't boiled, and then D through F are going to be the same as A through C, but instead of adding active lactase, you add the boiled lactase. So you can take a look at the methods, which will explain what is in each of these, but as you can probably see based on the color, B and E both have milk. Now before I added any of the enzyme solutions to each of the 6 samples, I wanted to make sure that each of the substrate samples were at about the same level, or basically no detectable glucose. So I took one of the test strips and tested each of the 6 samples, and as you can see they're all about the same color, this light bluish tint, and that's just the background coloration of the strips that indicates there's no glucose essentially that was detected. But I did want to take this moment to explain how the strips work. So the strips themselves have a few important chemicals in them. They have 2 enzymes. They have something called glucose oxidase. They have another enzyme called peroxidase, and they also have potassium iodide. Now when you dip the strip, if there's glucose present, it can react with this enzyme glucose oxidase to form gluconic acid, which is an oxidation of the glucose itself. And one of the byproducts of that is this H2O2, or hydrogen peroxide. Now the hydrogen peroxide can react with the peroxidase enzyme, also with potassium iodide, to form this I3-, or triiodide. Now the triiodide is what's going to give the color changes we're looking for. And it can give a yellow to brownish appearance depending on how much of it is present. And now if there's more glucose, that means you're getting more hydrogen peroxide and in turn more of the triiodide. And as you're getting more of this triiodide, it's causing the color change in the strip from this light blue to light green to dark green to a more yellowish brown. Ultimately it can be like a dark purple if there's enough of the glucose around. Okay, so the next thing I did was to begin the experiment with the enzymes. So what I ended up doing, instead of measuring out milliliters, because I don't have graduated cylinders or pipettes at home, I just did things in spoonfuls. So what I had done was add 2 spoonfuls of each substrate to each of the wells in my ice tray. And then I added to each, after I took that original 0-minute check with the strips, I added 1 spoonful of the enzyme to each. So A through C got 1 spoonful of the lactase solution that was not boiled, and then D, E, and F each got 1 spoonful of the boiled lactase. And then once I did that, I started my timer, and I took some glucose tests at 5, 10, 20, and 40 minutes after that initial addition of the enzyme sample to each of the substrate wells. And so now what you are seeing is the test results over time. So they were done exactly as before, where I dip the strips in the sample at the indicated times, 5, 10, 20, or 40 minutes. I lay them on the piece of paper, wait about 30 seconds, and then take a photograph. So that's what I did, and that's what you're seeing here. And so hopefully you can see some color changes happening, and your job will be to use this video, particularly this data, which by the way is also pasted into the lab document report that's posted on Canvas. So you'll have this available. You don't have to keep coming back to this video. But I would like you to record the results based on what you're seeing. So all you're really recording is the estimated glucose concentration in each sample, A through F, at each time point that's recorded. So I realized the images themselves are not don't have the time recorded on them, but that is indicated in the lab report document. So I have here this last picture is the image of the key, which has the color that corresponds to what glucose concentration you'll need. So good luck, and email me if you have any questions.